Review session, Monday night, 7 o'clock, the room that we were in last time, which is G32. We will meet there and do whatever you need to do. You've got airplane now. 5790. Almost everybody, we've gotten your data analyzed if you have data to analyze. If you don't have data to analyze, we obviously haven't analyzed it. Only one group that we don't have data analyzed for. The pollinators, we've run your data, and I believe you should have gotten an email scheduling time to meet with you on Monday during Joule time to go through the analysis with you so that you'll be able to put together your PowerPoint for next week. Next week is the exam on Wednesday, which means on Monday will be the discussion of the VNetL paper, so you need to bring your VNetL paper to class with you that day. Printed, name in the header, 2 pages, page and a half of summary, half a page of critical thought. The new guidelines are up on the iTunes U website, so it has formatting instructions, which for whatever reason they weren't in the last version. Just to remind you, 1-inch margins all the way around, 12-point font Times New Roman, double spaced, give us some space to write. And so if everybody's formatting it the same way, everybody has their name in the header, then 2 pages for one person is 2 pages for another person. Why are we limiting you to 2 pages? Actually, no, that is not the reason.
Supposed to be a summary, should include all the data and information.
Yeah, it forces you to pay attention to things that are actually truly important, and so it's actually a tool for becoming a better writer. What were you about to say?
So I had hoped to get all of your essay or first literature summaries graded and back to you in class today. They are well underway. I don't want to give some people back stuff ahead of time and other people, but I'm almost done. What I will do is, for those of you who used Turnitin to check your paper, I will make my comments on that so that as soon as I am done with everybody, I can just press go and I'll send an email and say, and your feedback will be on there. For those of you that did not use Turnitin to check your paper, I will mark on it by hand, but I'll just scan that in and send it to you so you can have that feedback, and you should have it by tomorrow before lunchtime.
What's really amazing though, and this was something that I read when I was an undergraduate taking a technical writing class, English professor did an experiment where he had 2 classes. They did the same amount of writing. In one class, he gave them feedback on every assignment and turned it back to them before the next assignment was due. The other class, he never gave them any feedback on anything. Both groups progressed the same amount during the semester. And he concluded that what makes you a better writer is writing a lot.
It's true, practice and writing. So what I noticed from your peer reviews yesterday is that not only are you better at peer review, but you're also better at writing. So I was looking at your writing samples and looking at the peer review that you all were doing, and it was extremely gratifying to see how much that you have progressed. On that topic, when you do get the feedback from the first literature assignment, please do read through that feedback. Part of the impacts of not getting feedback versus getting feedback, some people hypothesize that they are equivalent because there's not a lot of processing of the feedback that you do get when you get it. So what I would say is when you get that feedback, look at it, categorize the types of comments that are on there, and think to yourself, okay, what are the types of things that I'm doing that I could improve on? And kind of jot those down and keep those in mind when you're looking through your draft of VN et al.
And she writes tons on your papers.
I do. Writing improvement is important. I think it's one of the few things
not one of the few things, it's one of the most important things that we do here at Jewel that other places don't do, is really, really hone your writing. Bigger places just physically can't do it. Other smaller places don't put as much emphasis on it. It's one thing that employers, graduate schools, medical schools tell us that we do extraordinarily well, and they value it very, very much. They will seek us out to say, hey, we've had some graduates from your place, and holy mackerel, can they write. So pay attention to that feedback.
Plants and animals of the day. Grass today, Schizocarium scoparium. This is another C4 dominant grass. It's little blue stem, so it tends to have these bluish sort of stems in the same way that sometimes a big blue stem takes on a bluish or purplish hue. But this is a much shorter stature plant. It's once again a grass that comes up from a common root bunch. This will only get 2 to 3 feet tall, as opposed to big blue stem that can easily reach 7 or 8 feet tall, depending on the types of soils. It is one of those grasses that tends to dominate a prairie after you do nothing but burn it over and over again. So as you're reading Bean et al., and you see that the effect of burning is that grasses just kind of take over when you have burning in the absence of grazing. This is one of the species that is responsible for that takeover, as are Indian grass and big blue stem.
Dr. Klinsky?
Yes.
I haven't been to Konza Prairie. Is this the grass where it's like pretty super pokey, and if you, the edges of the leaves are kind of almost feathered downward, so that if you like brush against them, it's like, it'll slice you if you go really fast?
No, I do not know what that.
Okay. That is a grass that looks very much like that, but that grows in
Yeah.
southwestern Colorado.
Oh yeah, I know a grass like that in Puerto Rico. We call it sawgrass because it will cut you up, and we hate it.
Yeah.
No, this is not that grass.
Okay.
It will cut you if you take off a blade and try and make a little grass whistle out of it. You can slice your lip with it, but I have sliced my lip on Bermuda grass doing that same thing. So yeah, it's not anything like that. It has a really hardly visible flowering head that is typical of grasses, but it doesn't have that stereotypical turkey foot, that three-pronged head that you get on big blue stem. And it's also a much tighter grass than blue stem. It's much more dense coming up out of the bunch, out of the root mass. And it's the grass that my girlfriend is making me replace all of my big blue stem in my yard with, because she hates the tall big blue stem. This is my favorite prairie forb. This is lead plant, Amorpha canescens. It is a legume. You can see the pattern of leaves that are characteristic of the family Leguminaceae, Fabaceae rather. It used to be Leguminaceae, Fabaceae. These are the plants that fix nitrogen. They have these pinnately compound leaves. This particular species has these kind of silvery green, heather green sorts of leaves. It's very pretty in the springtime and summer when it's leafed out. It also has this tremendous flowering head inflorescence with lots of individual flowers on the whole flowering head. So because it's in the family Fabaceae, it's a nitrogen fixer. It's deciduous, and it's classified as a forb, but as they get older, they do begin to feel kind of woody to me, even though it's classified as a forb. And it will lose all of its leaves during the winter, but it doesn't die back like a bunch of annual forbs do. It will then leaf out the next year, and if you have one and it's growing year after year after year, it just gets bigger and bigger and bigger.
So Dr. Polinsky, is it considered deciduous because the stem does not die back?
It's deciduous because it drops its leaves. So deciduous,
Drops the leaves, okay.
deciduous is the term that we have for plants that lose their leaves during either in the temperate regions during the winter or in tropical regions during the protracted dry season. And it does that because it's not going to be doing photosynthesis anyway, so it might as well not be losing water and everything else. Oh yeah, and bison love this. It's one of their favorite
What
plants.
does pubescent leaves mean?
It means that they have pubes. They have little
Furry?
tiny short furry hairs
Yeah.
on them. Yes,
Okay.
the same root term. Sorry about that. Schizocosa ocreata. This is not your typical prairie animal. This is a spider. It's sometimes called the brush-legged wolf spider. You can tell it's a wolf spider because it has gigantic eyes. And then it's called brush-legged because the males have brushes on their legs, and they advertise at female spiders to basically prove to the female spider that, hey, I'm a member of your species, come mate with me. Don't eat me because you think that I'm a member of another species. The reason that I include this is that bison have an influence on plant community structure because they eat the plants. Spiders, there's a faculty member at K-State who has studied this. Spiders also have an effect on community structure of plants because where spiders are in high abundance, grasshoppers are in low abundance. And the effect of spiders in just being present and preventing grasshoppers from being in an area actually reduce herbivory on grasses, and so can have measurable effects on plant community structure, which is weird food webby kind of dynamic. They produce a lot of spiderlings. They're one of the species that carries their egg sacs, and tons of behavioral work has been done on them, not having to do with prairies, but with avoiding being eaten while trying to get lucky. And those are the plants and animals of the day. And I don't know where you are. Are you here?
It's Google Doc today.
Questions about any of that? All right.
That's a good question. So Taylor's asking, when they look at the prairie organisms of the day slides, and there isn't any information presented on the slide itself, do they need to look up? Are they responsible for finding more information?
No,
on-
just what we're talking about in class, although on many of these slides, if you look in the notes on the slides, there's much of the information that comes out of my mouth is in notes.
Right, so to see the notes, you'll have to open it
Open it
in
up.
the iPad version of
In
PowerPoint,
PowerPoint.
or on your computer in PowerPoint.
As you should always do with PowerPoints.
Okay, so the homework for today was to work on the worksheet that was relating C3 and C4 photosynthesizing plants to climate change. I had several people ask me, when you have a table like that, where it has something written on the left, and then it has a C3 column and a C4 column, what are you wanting us to put in these little boxes? Boxes are empty. I'm wanting you to put things that help you remember information on, like say, if this is water use efficiency, right? Which type of photosynthesis has a higher water use efficiency? C3 or C4? C4. And so when you're putting things in these boxes, you're going to put things that help you relate the two. And so you could put high for C4 and low for C3 if this was water use efficiency. Now, when you, I had spoken with a couple of you yesterday about some questions that you had about filling that out. I'm not going to go over the exact answers right now. I will post them, though, after class so that you can study them for the exam. One thing I think that will help, there was a question on the light efficiency of C3 and C4, who's more efficient at using light energy or harvesting energy from light. When we're talking about efficiency, we're going to compare two things, and we're going to say, for any single unit that we invest in this process, which one will give me more out? So we're looking for net gain. Okay, so the more efficient one will have a higher net gain, yeah? If you're comparing two cars, and you're saying which one has greater fuel efficiency, how would you define greater fuel efficiency in that circumstance?
Okay. Gallons per mile, how many gallons you input versus how many miles you get out?
Okay, so the miles per gallon, your mileage, your fuel efficiency is going to be how many miles can you drive per gallon of fuel put in. So for light efficiency, it's going to be how much energy net can you get out per input of photon. Okay, so think about, okay, for any given light that comes in, there's going to have to be an investment of ATP and ADPH, and you're going to get out glucose. And so when you're thinking about which one is more efficient, which one is more light efficient, C3 or C4, keep the energy inputs and the energy outputs in mind, and that should help you answer that question. Okay? All right, so our learning goals today, we have kind of a lot to talk about. We are going to work on our diagram of the pools and fluxes of the nitrogen cycle in Konza Prairie. Okay, so make sure that you have access to that drawing that you were making. This is where we were yesterday, or Monday, I guess it was. Okay. So, on Monday, we had defined some of the pools and fluxes in the nitrogen cycle in general, and specifically at the Konza Prairie. And we will add to this image as we go, right? We have soil, and it functions as the place for several pools of nitrogen. Urea, dead organic matter, microbes, plant roots. We have groundwater as a pool of nitrogen through it basically starts leaking out of the soil. It's called leaching. We'll talk about it today. Animals are a pool. Plant shoots are a pool. The atmosphere is a pool, right? And so we're going to add to this image as we go. Remember that nitrogen fixation is the process by which this atmospheric nitrogen, the N2, is converted to ammonia. It's carried out by bacteria. That bacteria can be free living in the soil, or it can be in association with plants. The bacteria can also be photosynthetic, or it can be heterotrophic. And that nitrogen fixation makes nitrogen available to plants for the production of amino acids and other things that contain nitrogen. Remember this image, we've got our nitrogen fixation in association with plants. The bacterium infects, makes a nodule, and once that is large enough, they have an area of that nodule that undergoes nitrogen fixation. We said this is a symbiotic mutualism. Okay, so symbiosis, what does that mean?
Two organisms living in the same space together.
Usually one is acting as a host. Okay. What is mutualism? Chris?
Both organisms benefiting.
Both organisms are benefiting. You want to add anything? No? Okay. All right. Okay, so in this symbiotic mutualism, what are they getting? Turn to each other and talk about what is the plant getting, and what is the bacterium getting? I want to hear lots of talking. What's the bacteria getting? Talk to each other. What do you suppose it's getting? If you don't see them on there, within an hour after class.
In this case, the
Okay. So what is the plant getting? That one's probably a little bit more obvious, right? What's the plant getting from this relationship? Nitrogen, right? It's getting nitrogen in a form that it can use. Okay. All right. So the plant gets ammonium. What is the bacterium getting? Any ideas? Asa? Pardon? Shelter. Absolutely. The plant is getting protected from the environment. It's inside that root nodule. It's not going to dry out. It's not going to get eaten. It's protected. Okay, what else is the microbe getting? Connor, if you had to guess, what would you say?
Maybe like resources, kind of. I mean,
Yeah, resources. Keep going.
you said it lives in the root.
It lives in the root, yeah.
Yeah, so we'd be getting like water and everything in the plant.
Could be getting water, yeah. They can, if the plant is getting water, if the microbe was in the soil, it probably could get water as well. But let's keep going with this because you're totally on the right track with resources. Okay. Other ideas? Claire, what do you think?
So it's like the bacteria, so the plant is like a host.
Yeah.
So we get food.
Absolutely. These nitrogen-fixing bacteria that live inside the roots are getting their food from the plant. They're actually eating the sugars produced by the plant. So they're not, the ones that are living in the root are not photosynthetic. They're not eating stuff from the soil, right? They're inside the root. They don't even have access to the soil anymore, right? They're not eating the plant. If they were eating the plant, that would not be a mutualism. That would be a parasitism. So the plant is actually giving them the carbs that the plant makes. So the plant will make starches and simple sugars. Okay, awesome. And the bacterium is protected from the environment by the host. Okay. So this is just an example of some data that were collected by Becker and Crockett in 1976. It's been a long time. I was like less than one probably when this was, and I'm ancient, so that tells you how old this is. So here's the plant weight in milligrams. This is the micromoles of carbs produced by the plant per day, carbon compounds. This is the nodule weight, so the weight of that little place where the bacteria are living. Okay, and that nodule, the size of that nodule is proportional to how many bacteria are in there. So this is a good proxy for how much the bacteria are growing, how well they're doing. As you can see, as the bacteria do well and the nodule gets bigger, the plant weight is also going up, and the amount of carbon fixation that it can do is also going up. So we have this nice mutualistic relationship. You guys have questions on that?
this is not about the nitrogen, but look at that figure. Those of you who have done data analysis in lab, what do you notice about this figure? It is drawn by hand.
That is true.
It is drawn by hand, hand lettered, all those little dots. This is what people did in the 1950s, 60s, and 70s, and into the early 80s.
The numbers may be typed.
I think the numbers are dry transfer.
Oh, yep,
But the
I bet you're right.
lettering.
But the lettering is absolutely by hand,
handwritten,
yep.
And the little dots and everything is handwritten, and you can see the inconsistencies in the axes that also indicate that that's all done with ruler, pen, and paper.
It's a little bit longer on that side than it is on this side.
It's a little wobbly.
A little wobbly.
Anyway, just technology has changed a lot even since
It has.
this time period.
In the, in, let's see, 97 is when I was really starting to get into biology research as an undergraduate. And it was right at this weird transition where most of the research that existed, you had to go get it either on paper or on microfiche. Have you guys ever looked at microfiche before in the library? There are these crazy machines, right? You have.
I have, oh, yeah.
I was kind of not wondering that question about you. I think I maybe knew that answer. They look like, gosh, how would you describe them? Very old, like TVs with like a built-in desk, and you plug in like a little cartridge, and it makes this crazy noise when you advance it. And it's like right when it changes to the next thing that you look at. And it's basically like somebody took a picture of what it was and then made a full-on slide, like you would put in a slide projector kind of. And that's how you had to find your stuff. But it was right on the edge of when things were starting to be available online in PDF form. So it's kind of crazy when you go to look for papers during that time. They either look like this and look weird and blotchy, or they look like super nice, and it just depends on, you know, like what journal it was. All right, so given that we've got this nitrogen fixation that's happening, there is some happening in the soil, but I want us to think about the nitrogen fixation that's happening in the root nodules of plants. What is limiting nitrogen fixation in this system? Think about it for a second, and then I want you to talk to each other about it. Because all the aspects of the answer to this aren't necessarily super obvious. Okay, what is limiting nitrogen fixation?
They can't use just like nitrogen that's around. They don't Yeah. Yeah. So they can kind of taste it in, but it can't do anything with it until it's been like. So I guess that's
All right, what do you guys think is limiting nitrogen fixation? Okay. All right, what do you guys think is limiting nitrogen fixation in this system? Four fast, four curious.
Talk about phenol and less in the area, possibility, possibility, like the excretion, the dying, such nothing issues.
Okay, so I'm gonna try to summarize or rephrase your answer and tell me if I'm on track or not. Availability of nitrogen just in the system. Okay. Cool. All right. Okay, tenacious turtles. What do you think?
We said the amount of ATP available.
Amount of ATP available. Okay. And where is that ATP coming from? And to who, available to whom?
One's coming from photosynthesis and available to nitrogenase, the enzyme that catalyzes the reaction.
Awesome. Okay. Amount of ATP from photosynthesis available to nitrogenase. Okay. Awesome. Let's see. Jack, what do you guys think is limiting photosynthesis? Nitrogen fixation in this system.
Photosynthetic rate, determining the amount of ATP.
Sure. I'm gonna put this kind of above that because the amount of ATP is dependent upon it. Absolutely. Chaperones. Anything else you think that's limiting nitrogen fixation in this system?
We talk about air,
Air. Okay, so is nitrogen fixation an aerobic or anaerobic process? That's a good question.
I think because it's fixing atmospheric nitrogen, and two, it has to have access to air.
Well, it's inside the root. Is the root in the air? No. But I know I get where you're going with access to nitrogen, but I think that's probably just availability of nitrogen. I think would go along with that. Look back in your notes. Is nitrogen fixation an aerobic process or anaerobic process? Pardon? Anaerobic. Anaerobic. Yeah. Okay. So let's see. Photosynthesizers. Is there anything else that you want to add to limits to nitrogen fixation? There is more to add, so if you want to take a guess. No, because remember this is an anaerobic process. Well, yes, no, it's an anaerobic process, and oxygen is going to limit your photosynthesis. So I guess if there's too much oxygen in the atmosphere, that might be bad. So that's going to go back to photosynthetic rate. That's good. All right. So you guys got most of the obvious ones. So in this system, the first thing that's going to limit nitrogen fixation is, I think you guys nailed something that I didn't actually put on here, presence and quantity of bacteria. Okay, so you've got to have the nitrogen fixing bacteria present. Totally agree with that. You do have to have availability of nitrogen to to fix. Okay? So if your soil is super depleted of nitrogen, you're not gonna be able to fix it. So even though I don't have that on my list, we should add these two. These are important. And this one particularly, the availability of nitrogen to fix, not always a good assumption to make. There might not be nitrogen to fix in that system. There might be nitrogen poor because, for example, in agriculture, if you don't use a ton of fertilizer and you plant a crop that is not nitrogen fixing for many years in a row on the same plot.
But that's nitrate. That's nitrate that's getting depleted. I would think that the availability of nitrogen in soil is directly correlated with the availability of nitrogen in the atmosphere. And all of the air spaces, so I would say seldom is that into because I'm thinking into.
You're thinking slowly into.
Yeah, because if it's already in the form of nitrate, then it's already been fixed by somebody. Because this is about infixation. Am I
Yes.
mistaken something?
So then how would you explain then agricultural fields that are,
Depleted nitrate. They just take up the nitrate year after year after year, and as long as we're not putting in a nitrogen fixing species or supplementing with synthetic fertilizer that has nitrate in it, you're depleting nitrate, but N2 is in the air spaces in the soil. It's just not available to plants, and that's unless I'm.
no, I think you're right. So I'm thinking of, the way I'm thinking of it is that if you have no, I guess so then it wouldn't be the availability of nitrogen fixed would be in the atmosphere. What I was thinking of is the, yeah, you're right.
The health of the plants.
I was thinking of the presence of these guys to keep up with how fast it's being, the nitrates and nitrates are being used in the crop. So you are.
Yeah, I think I would probably just cross that one off because soil air is different than atmospheric air, but usually it's just mainly different in that there's less oxygen and more carbon dioxide. I don't know that nitrogen changes that much comparing soil air and soil interstitial spaces to atmospheric air. What's the most dominant thing in atmospheric air? You breathe it in, breathe it out every day.
Nitrogen remains the same.
N2, what's the percentage of the atmosphere that's made up of N2? 78%. Seldom is N2 going to be limiting in any reaction.
You're right. I was thinking of it in the
terms of
wrong way. I was
nitrogen
thinking of it in the form of the rate of nitrogen fixation replacing fixed nitrogen.
So if this is an
in the system.
N2, then probably.
Right. No, you're right. Thank you. Okay.
So the reason you didn't have it up there is it probably shouldn't be up there.
It's because it shouldn't be up there, right? Okay.
Because I challenge you to find that as a limiting factor in any textbook on nitrogen fixation.
Yeah, no. I can't
No.
think of any circumstances in which
It
it
is,
would be.
it is interesting though, if you fertilize a field over and over and over again, and then sample that field for nitrogen fixing bacteria, there aren't nearly as many of them, because it turns out, if there's a lot of nitrogen, nitrate in the soil, plants that would normally give up some glucose to bacteria to fund the production of nitrate for them, they stop doing that.
Yeah.
And so that's another way in which agriculture can do things. The same thing is true of mycorrhizal fungi. If you have a lot of phosphorus fertilizer being added to a site, the plants basically can absorb that sufficiently without the presence of mycorrhizal fungi. So they just stop giving up glucose to mycorrhizal fungi. Diversity and abundance of mycorrhizal fungi go down. And then if you ever stop adding fertilizer, they have a harder time because they don't have those fungi in the soil.
Being a host is costly, and so unless you're
It
getting
is.
something out of it, you really want to combat that relationship and not be a host. Okay, so limits to nitrogen fixation, light, we need energy. And one way that energy can be limited is sunlight availability. What just happened? Here we go. Sunlight is going to give us ATP and NADPH. In what part of photosynthesis do we get ATP and NADPH produced by the conversion of light energy? What part of photosynthesis is that? The light reactions. Yeah, absolutely. And in order to make ATP and NADPH, we also need phosphorus. Okay, so just to make NADP, we need this P stands for phosphorus. Just to make NADP so that it can then carry an electron and be a high energy electron carrier, we need phosphorus. In order to make a DP, which is the precursor to ATP, we need 2 phosphate groups, and so we need phosphorus. And in order to energize or carry that energy, we need a 3rd phosphate group to make ATP. So limits to energy here are sunlight and phosphorus so that we can convert that light energy into chemical energy carried in the form of chemical bonds between phosphate groups or as energized electrons in NADPH. Okay, so just as review, energy coming into the system can limit nitrogen fixation. The 2 elements of energy limitation can come in the form of sunlight and phosphorus. Okay? In Reed et al. 2007, they did a study on nitrogen fixation rate, and they studied how the presence of additional phosphorus, no additional phosphorus, or removing a little bit of phosphorus, what effect that had on nitrogen fixation. There was no significant difference between removing phosphorus and having the control. They couldn't really get rid of a lot of the phosphorus. It's hard to do. If they added phosphorus, suddenly you have a much higher fixation rate, nitrogen fixation rate, because the presence of phosphorus is no longer a limiting factor, has tons so that it can make ATP and NADPH. Nitrogenase. Oh, question? Yeah.
The energy is by the plants or by the bacteria?
By the bacteria and by the plants, right? So the plants are using ATP and NADPH for what? They're from photosynthesis, but what are they using it for? Right, so they're using it in the Calvin cycle to make glucose. Okay, so the nitrogen fixing reactions use ATP and NADPH, but they also use, and some of that is produced by the light reactions. There's also some ATP that is produced by breaking down glucose, right? And the bacteria are respirating that. They're breaking down that glucose and making ATP, right? So the energy is needed by both the plant, to keep the plant alive, but it's also needed by the bacteria. And when we're talking about limiting nitrogen fixation, we're really talking about direct effects on the bacteria, because that's the thing that's doing the nitrogen fixation. Does that answer your question? Okay. All right, so the other thing is nitrogenase. What is nitrogenase? You remember. I know you do. It ends in ase, so we know it's what kind of thing? An enzyme, okay? And the enzymes that we've studied in photosynthesis so far, what do they do? They're catalyzing a reaction. Good. Okay. And nitrogen fixation isn't included in photosynthesis, but using photosynthesis as an analogy, what were the enzymes in photosynthesis doing? In addition to catalyzing a reaction, what kind of reaction was that? A fixation reaction, right? So they were taking an element and incorporating it into a biologically useful form. So what do you suppose nitrogenase is doing? Fixing nitrogen, right? It's taking that N2 and it's doing this really costly ripping apart of the N2, using a bunch of energy to do that, and then putting that nitrogen into a form like ammonia or ammonium or nitrate, so that the plant can use it. The bacterium also can use it in that form. Nitrogenase is made from a bunch of sort of common stuff that's around, but one of the elements that is less common and used in fewer organic reactions is molybdenum. This is the one that I told you I will always say slowly because it's hard to say. Iron and sulfate are also components of nitrogenase, but they're quite a bit more common. If you restrict molybdenum, you can't make a ton of nitrogenase. And so here's Wurzburger et al. 2012. We have ARA, which is basically nanomoles of these carbon that you can make through photosynthesis. So this is, let's see, molybdenum is added and phosphorus is added. You get loads and loads of nitrogen fixation, which leads to a good photosynthetic output. If you have molybdenum but no phosphorus added, you're like, meh. If you have no add extra molybdenum, but you have phosphorus, eh. And then if you don't put any extra of either, eh. So you really need both. So we're limited by energy, we're limited by nitrogenase. Energy limitations are availability of sunlight and phosphorus. Nitrogenase limitation, availability of molybdenum. Questions? All right, we're going to go back to our diagram of pools and fluxes. Okay. Remind me what this diagram should look like. What's the big box? Largest box. Soil. Okay, so we've got a big soil box. Okay, what else do I need? What's down here?
Bedrock.
Yep, this is rock.
Groundwater.
Groundwater. Is this a pool or flux? Pool. Okay. What do we have inside the soil? What kinds of pools do we have in here?
Microbes.
Microbes.
Plant roots.
Plant roots. What else do we have?
Excrement.
We have excrement. I'm going to call it dead organic matter. Okay, what else do we have? Pardon? Animals. Okay, so animals not in the soil yet until they're dead organic matter, but they're above the soil. We've got animals. Okay. Also in the soil, we've got urea, right, from those animals going to the bathroom. Okay. What else? What other pools do we have? The atmosphere. Okay, those are the ones we mostly talked
Plant
about.
shoots.
Whoop, plant shoots, thank you. Almost forgot those, my goodness. Okay, so, all right, we've got some nitrogen inputs into this system. So we're gonna draw those fluxes now. We've got, y'all talked about atmospheric deposition, right? So we've got wet falls, rain, snow, fog, clouds. We've got dry fall, which is just like particulates, right? Aerosols. So atmosphere. There's a flux there. And how does atmospheric N2 get into plants and plant roots? You guys know this, we just talked about it. How does the atmospheric nitrogen get into the plant roots?
By a microbe.
Yes, and that process is called give you a hint, it starts with nitrogen. Fixation. Nitrogen fixation. Okay, so this is a flux. Atmosphere to plant roots. Nitrogen fixation.
Well, my point, wouldn't the flux be from atmosphere to microbes and microbes to roots?
Yes. Let me. So some microbes that do nitrogen fixation are free living. And so why don't we do this? I'll put a little M inside the plant root. Does that work? Question?
Lighting be a factor.
Can lighting be a factor
Yeah, it's
in fixing like
Oh,
nitrogen?
yeah, lightning fixes a little bit of nitrogen.
What?
Lightning fixes a little bit of nitrogen.
Oh, lightning. I thought you said lighting. Like, I'm not like lightning. I do not know the answer to that question.
It does.
It does?
It does.
Okay.
It's teensy
Okay.
compared to biological fixation and human fixation.
Okay.
That's a technical term, teensy.
Teensy weensy. All right. Humans have increased nitrogen fixation about threefold. So we plant nitrogen fixing crops. For example, I grew up in the pinto bean capital of the United States, which is my claim to fame. We do a lot of fixation via fossil fuels emissions, which makes nitrogen particulates in the air, and so we get nitrogen deposition through dry fall. We also do industrial nitrogen fixation via this Haber-Bosch process, which is basically like we make artificial fertilizer and we pour it all over our crops. And that's a major input of nitrogen into the system. However, we're drawing ponds of prairie here, and so those human inputs we're not going to indicate on here, with the exception of the dry fall from the atmosphere. You know, it's not an isolated system. Okay. All right. We're going to talk about some nitrogen transformations, because if you remember, when you go from one pool to the other, when you have a flux or an arrow, it can be just a change in, it can be a physical change, it can be a physical transformation. Okay? So we're going to talk about some of the transformations. Oops. We can have, so litter fall, so when leaves fall, we have live organic matter becoming dead organic matter. When you have branches that are ripped off of trees from storms, when you have fall come along and the leaves dry up and most of the nutrients actually are resorbed by the tree, but some of them are not. We have dead organic matter that falls to the earth. And so that is already on our diagram here, the dead organic matter.
Are you counting feces as part of dead organic matter?
I am. Yep. So we can even put little examples on here if you want. So we can poop, leaves, dead animals, stuff like that. All right, so we've got dead organic matter, and that's one form of nitrogen. You'll have all kinds of different forms of nitrogen in something that's dead because it used to have lots of forms when it was alive, right? All right, so but then we also have decomposition, where that dead organic matter is turned into dissolved organic nitrogen. Okay, so this is basically when the dissolved organic nitrogen, or the nitrogen itself, through decomposition, oops, that's the wrong color if I'm putting my fluxes in blue. So dead organic matter can produce dissolved organic nitrogen in a couple of different ways. Through decomposition, which is what I'm gonna put here. So that's the main flux into dissolved organic nitrogen that we see in the Konza Prairie. And I'm gonna abbreviate that DON, okay, dissolved organic nitrogen. But it also can include leaching into groundwater. Okay, so that litter fall or dead stuff its nitrogen can leach into groundwater. When you just fragment nitrogen compounds, this can involve changes in physical and chemical structure, ingestion, and excretion. So there is some dissolved organic nitrogen that is in meat. Okay. Question? Fragmentation. Like fragmentation would be basically the same as changes in physical structure. So it might be like a large protein that is actually just broken, right, into smaller proteins. Do you want to add anything to that, Dr. Polinsky?
It happens at larger levels, so.
Yeah. I mean,
So whenever leaves are on the ground, they will be fragmented by a whole suite of decomposing organisms. They're usually some type of arthropods, breaking them into physically smaller portions, and that actually facilitates microbes then getting access to those because it increases the surface area of those things. So it's more of a physical process rather than a chemical process, the way ecologists use it.
right. And so I would not include chemical breakdown
I wouldn't either. It's strictly physical.
in physical. This is not changing the chemical structure. Okay. Major decomposers are bacteria for animals. And fungi are mostly broken down, plants are mostly broken down by fungi. For those of you who may notice that once the leaves start falling, your nose will not stop running. It's probably because you have an allergy to the fungi that decompose leaves. I am in that category. The rate of decomposition is influenced by temperature, moisture, plant litter quality, soil disturbance. These things make a lot of sense if you're thinking about, okay, well, decomposition is being done by a live organism, Live organisms require particular conditions for doing well and for growing. All right. We've got ammonification or mineralization. So this is going to have some examples. So this is the process by which that dissolved organic nitrogen can be turned into ammonia or ammonium. Okay, so, again with the wrong color. Sorry, it's dark. Okay. Let's see. I'm going to add a flux here that is plant death. I am missing a slide, gosh darn it. Okay, for ammonification and mineralization, microbes will take up that dissolved organic nitrogen and secrete ammonia if it is in excess of the available carbon, and that's rapidly converted to ammonium. Okay, so that's controlled by the availability of the dissolved organic nitrogen. If there's none there, it's not going to happen. It makes sense. It's controlled by the availability of microbial carbon to nitrogen. It's controlled by temperature and moisture because the fungi or the bacterium has to be happy, right, to do well. All right. So my question then is, how would an increase in CO2 in the atmosphere affect that nitrogen fixation through ammonification? How do you suppose an increase in CO2 in the atmosphere would impact nitrogen fixation? Caitlin, what do you think? Okay, let's go with that. Okay, let's imagine, okay, so maybe it'll increase, okay? So CO2 goes up. Walk me through the logic of what would happen.
So if the CO2 goes up, then more diffusion of CO2 is taken into the leaves, and so then the rate of photosynthesis would increase.
Okay, which would?
So if photosynthesis increases, then you have more energy being made that would be available, and so if you have more energy being available, then
Okay, that is a very logical route to take. And that's maybe what you would predict, except that the microbial carbon-nitrogen ratio will determine whether the fixation happens. And so if there's more carbon in there, it actually starts to make it where nitrogen fixation doesn't happen anymore. Okay. All right, so I know we're out of time. So what I'm going to do is there are just a couple of more slides. I'm going to post them all with some explanation. So that and the slides for Monday will be available. And I'll also have a diagram, okay? The Kanza pools and fluxes diagram. Sound good? Questions before you guys go? Oh, homework. Turn in your homework.